row of differentiation, they're actually the target of many, many drug development programs related to many different diseases where these proteins could go awry, or where you could use it as targets for infectious disease. So the GPCR class alone, which is probably the most important class of receptor proteins in our body, there's a number of blockbuster drugs from various companies that treat various diseases. So what's the measurement science that need to be addressed here? So again, anybody who's worked with membrane proteins, this is not an unfamiliar list. The first issue is making functional protein. That's not something that we focused on, but it's really, I don't like to leave that away, because that's really critical to actually being able to really enable studies of these kinds of proteins. The other problem, which is now somewhat being not quite front and center any longer, is these proteins are functionally dynamic. So there's now a lot of beautiful crystal structural data. This is, for example, the crystal structure of the adrenergic receptor with its G protein bound, covered nature. But these proteins are actually not static. And so dealing with the fluidity of the membrane, which these are, again, done in detergent solubilization form, so this is sort of a cartoon. So you have to understand their full function. You need to understand how they're dynamically moving, in addition to what these sort of snapshot crystallographic pictures can show us. So the other thing is detergent solubilization can sometimes trap a state that's not particularly active. And oftentimes, to get crystal structures of these proteins, significant engineering goes into the proteins where they add domains for stabilization. They crystallize them with antibodies to, again, stabilize the proteins. And so you don't have the true native state of the protein in the bilayer. Okay?